All right, I'd like to welcome everybody. Thank you for being here. We're really excited to get started. So I have just a couple of housekeeping things I'd like to go through before we turn it over to the speakers. Please submit your questions through the Q&A bar. We will answer them, as we go through, and then there will be Q&A at the end as well. If you require a continuing education certificate or a certificate of attendance, there will be information in a follow-up email at the end that will tell you how to obtain one of those. And then finally, the webinar is being recorded, and within 2 to 5 business days, we will have it available for review on our YouTube channel. All right, I'm going to go ahead and turn it over to Dr. Brian Holub. Thank you.
Hello, can you hear me now? Well, I am Dr. Brian Holub. I'm chair of the Scientific Review Committee and on the board of directors for the Every Cat Health Foundation for many years. Every Cat has been funding FIP research at the top institutions throughout the world for more than 40 years. We've been at the forefront funding more than 60 studies, ranging from such things as understanding the disease in the 1980s to effective real-time reverse transcriptase PCR testing, to curative therapies culminating in Dr. Niels Peterson's landmark paper published in 2016 on GS 441524. So it's been thrilling to shepherd research for this disease. It was once considered 100% fatal, and of course now we consider it curable in most cases. The research continues still today with research to understand other effective therapies, a new understanding of the basic biological features, and maybe even changes to the basic disease process. Every Cat has hosted numerous webinars and international symposia on FIP, sharing the most current information with our audiences. Supporting research is only possible with the dedication of individuals like those of you joining us today. Currently, one of Every Cat's greatest supporters in the fight against FIP is Zen by Cat. Thank you, Peter Cohen, for helping to highlight this webinar today. Our deepest gratitude to everyone helping with efforts in cat health. As soon as Every Cat became aware of the situation unfolding in Cyprus, we quickly responded to researchers and to become one of the first organizations to approve an initial study, which we funded last year on October 17, 2023, in a special emergency funding grant cycle. And it's now my great pleasure to introduce one of those researchers.
Dr. Gunmore, go ahead.
I missed that little bit. I'm so sorry to miss that last bit of Dr. Brian's introduction, but the work that Every Cat does, we don't need to say, do we? That's why we're here. And we all know that it is so important. So that is why we're going to do this webinar panel discussion for you today. I've been lucky enough after meeting Haralampos Epita, and he's not here today, but we've got everybody else who is working with him, and he brought the data to me, and I just looked at it because someone pointed out, brand new member of staff, and he said, who does anything about FIP here? And someone went, go see Danielle. And he showed it to me and said, should we be worried? And I went, and from that, it's spelled out, and we're being able to do so much important work for this outbreak. So we're bringing together 3 essential people for you today. We're going to start with discussing the epidemiology of how it was first seen and how it has developed. That is Dr. Dimitri, I'm going to try and get your name right, my love, Epinandinos. Oh, I'm so sorry. I'm so bad at languages. Epaminondas, I think. He'll correct me, please do. I get my husband's name wrong half the time and call him one of the cat's names, so, you know. Then we're going to have Brilliant. Christine Tate-Burkhardt speaking. She's going to be talking about the sequencing and how she came to discover just what a scary monster that we've got, you know, a real Frankenstein's virus. And then we're going to finish off with Maria Liraki. She is about to start a PhD. Actually, Dimitri is starting a PhD on this as well, as is Maria. So Dimitri is going to be looking at the epidemiology in a lot more depth, and Maria is looking into the therapies and interventions as part of their PhD programs. And Christine, of course, is hosting all of the virology and sequencing. So I'm going to shut up and let Dimitri, Dr. Dimitri, are you going to come and play with us?
Okay, and sorry, correct. So, thank you, Professor Gunmore, for the introduction. Good morning or good evening, everyone, depending on which part of the world are you watching us from. I would like to thank Every Cat Health Foundation for giving us this opportunity to share our knowledge from this feline infectious peritonitis outbreak. This presentation will be an epidemiological journey through this FCoV23 outbreak. The topics of this presentation will be introduction of how this outbreak was introduced in Cyprus, our action plan as in Cyprus, the publicity of this outbreak, the collaborations for research purposes, and some epidemiological data of this FCoV23 outbreak. First of all, let's say a few things about Cyprus. So let me open my pointer. Cyprus is an island in the eastern Mediterranean Sea, which is known since the ancient times for its mineral wealth, actually about copper. That's why our color here in the flag is the copper color. We're also known about the most ancient documented wine so far, which is Commandaria, and we're also known about our sea and about the sun. We are the eastern border of the European Union. We are a full member of the European Union, and we are sitting somewhere here, between Europe, Asia, and Africa. We are also known as the island of cats because of the high feline population in the island. And a very interesting fact is that the oldest known pet cat was discovered in a burial place that was estimated to be 9,500 years old. Now let's go to the outbreak. On December 22, the increased number of suspected FIP cases were reported from colleagues from all over Cyprus. And this alarmed us, our association, and we have prepared a questionnaire in order to gather more information about this concerning outbreak. The questionnaire covered the years 21, 22, and the first months of 23. This is the questionnaire results. You can see this graph here. This graph has two different colors. The red color that represents the number of suspected cases based on FIP clinical criteria, and the orange color that has represents the number of confirmed PCR cases. All those data were collected from colleagues. You can see the trend of the increased cases from 21 to 22, and you can see that there was a very big increase on 23, on the first 3 months of 23, actually 4 times than the whole of 22. This was an alarming situation, so we have to come up with a plan. The first thing that we did was to inform the authorities. So a letter was sent to the director of the veterinary services about with which we were closely collaborating throughout the outbreak. And the second part was informing our colleagues, the veterinarians, and the public. About the veterinarians, we have delivered 3 webinars for the vets, and that was done to have a uniform approach on the diagnosis and the treatment of FIP. The other, we have frequently posted with media posts and made media briefings for the public, and one webinar was dedicated to the animal custodians about FIP. We have raised this matter to the Federation of the Veterinarians in Europe and the Federation of European Companion Animal Veterinary Association, which are these 2 bodies of the veterinarians in Europe. And the last thing, which is was the most important, was the formation of a scientific task force with the University of Edinburgh and the Roslin Institute. As Professor Gunn-Moore told you, Haralambos was the link between Edinburgh University and our association, and this collaboration brought the first publication about this concerning outbreak. We have issued a publication to the Veterinary Record. This is a journal of the British Veterinary Association, on which we have declared that there is a concerning feline infectious peritonitis outbreak in Cyprus. That had a massive effect on the publicity of this outbreak, and this massive effect was, in marketing terms, it went viral. Actually, it was a virus outbreak that gone viral, and a lot of important organization has reshared this article with the rest of the public. This is FCoV, the International Cat Care, a very known organization about cats, the British Veterinary Association, of course, Every Cat, and some newspapers, like The Guardian and the Washington Post, have mentioned this outbreak. This massive publicity of the outbreak brought a lot more collaborations. A lot of institutes and universities wanted to research this, the reason of this outbreak. We had the opportunity to have Every Cat Foundation investigating this outbreak and funding a big part of this outbreak. And because of that international concern of this outbreak, and the institute wanted to help with this outbreak, we have formed an alliance, as we call it. This is a universal alliance to research the FCoV23. We have called it the Center of Excellence, and this is a very big opportunity to research this outbreak. Now, the questions that has to be answered through the research. The first question was the genome sequencing of the entire virus and the phylogenetic analysis. To record the epidemiological data of this outbreak, to record clinical data and the characteristics of this outbreak, find new diagnostic tools to help with the diagnosis, record data on new and existing treatment options, and this generated data could possibly aid for the design of a new vaccination for FIP. But why did we give this name, FCoV23? We will talk about a bit for the molecular epidemiology. The PCR testing revealed that the same strain in all the PCR-confirmed cases. These cases clustered together, suggesting a new FCoV variant that causes the clinical appearance of feline infectious peritonitis, and that is the very important finding that we had. And there was a very strong indication of direct cat-to-cat transmission. These all were stated in our preprint that was issued on November. This is what Dr. Buchert will talk to you about in the next presentation. Now let's focus on the epidemiological data of this outbreak. Here you can see the PCR positive cases that were submitted in a single laboratory in Cyprus. Cyprus is a small island, so we only have a single laboratory for companion animals. So that data went, all the data went to this specific laboratory. You can see that the PCR confirmed cases, there were low numbers of PCR confirmed cases during all 2022. but you can see that there was an increase on January of 23. That was the time that this outbreak has started. On the big graph here, you can see the PCR-confirmed cases divided on from January 23 till July 23. This is 8-month period, and you can see the district distribution of these PCR-confirmed cases. So the outbreak has started from the capital city of Nicosia, in which January there was an increase of the cases, February was a peak, and then there was a decrease on the PCR positive cases, but the other districts started having increased cases. You can see this in this map here. This is the map of Cyprus. This is the capital city of Nicosia, which the outbreak started. This is Larnaca, Famagusta, Limassol, and Paphos. You can see that in the timeline, this is January 23, the PCR-confirmed cases were increased in Nicosia first, peaked on February, and then the other districts started having cases of PCR-positive cases of FIP. The epidemiological monitoring was done in 2 ways. The first way was the one that I showed you with the PCR-positive cases, and the second way was due via questionnaires. So our association delivered another 2 questionnaires, one that in the 6-month period, so it was launched in July, and the third one was in December 23. I will present you some data from the last questionnaire that we have issued, and this questionnaire gathered data from our colleagues in Cyprus. And as you can see, it have put a timeline, the evolution of FCoV cases, FCoV23 cases from January 22 to November 23. As you can see, there was a stability in the cases on January, but there was an increase of the cases from the 4th trimester of 22, peaked on the second trimester of 23, sorry, and there was a decrease of the cases, and now the cases are stable in a number. We have launched another questionnaire for the collection of the clinical data. This was prepared from Dr. Maria Liraki, which is the next one of the speakers today, and those clinical data were from this questionnaire that was presented from here. The most interesting clinical data that we have gathered within epidemiological meaning are that cats of any age could be infected, in contrast to the existing literature about FIP. And the most clinical appearance is the wet form, but there was an increased representation of neurological cases in this outbreak. About the treatment options now, as Cyprus, we are the first European country which has approved 3 different pharmaceutical products for the treatment of FIP. The treatment were approved in accordance with the European Medicines Regulation, and these are the 3 different options. It's injectable remdesivir, which is a veterinary formulation. This is the GS 441524, which is an oral medication. Again, it is a veterinary formulation, and this is Lagevrio. It is best known. It was approved for COVID-19 during the COVID times. It's Molnupiravir, and that was used for these cases in Cyprus. Our future investigations will be focusing on the monitoring of the epidemiological data on FCoV23 outbreak, about the investigation of the epidemiological characteristics of FCoV23 variant. For instance, alternative route for the infections may be possible, so we will research on that, and the environmental shedding of the FCoV23. And we will collect data and investigate the epidemiological characteristics of FIP and FCoV in the neighboring countries, because we have a strong indication that might be this outbreak came from another country. Okay, this is the list of acknowledgements, the list of people that I have to thank about for this work that they have done. It's enormous. I picked a few of them to thank because they were really important for this research work. I want to thank our veterinary services for the collaboration. I want to thank Dr. Maria Liraki, which was great in this, which was great with this outbreak. I want to thank the University of Edinburgh and the Rosling Institute, and especially Dr. Karalambos Atipa, Professor Daniel Ganmour, Dr. Stella Mazzeri, Dr. Christine Tate Booker, which are the presenters here as well, for the work they have done so far. Our president, Dr. Nectaria Ioannou Arceloglu, the laboratories that have worked together, the FCoV23 Center of Excellence, which is a scientific union actually for this research, and it's really important to do that. EveryCat Foundation, of course, and the last addition was Dr. Maria Eleni Filipidzi from the Aristotle University of Thessaloniki. Of course, I will be very ignorant to not to thank our colleagues in Cyprus. They have done a lot with so many low tools that they have on their hand. They were really great managing this disease, this outbreak, and I want to thank them so much. And of course, my family, because I have neglected them so much in this past few months. And last, I would like to invite you all to a very special Euro Congress. This is one of the most important Europe events, veterinary events in Europe. This is the 29th FCoV Euro Congress, which will be held next September in Athens, the beautiful city and historic city of Athens, Greece. And we will be there with Dr. Maria Liraki to present another fact of this FCoV23 outbreak. Thank you so much, and sorry for taking more of my time, Dr. Ganmour. Thank you very much.
I don't think that was a problem at all. Thank you. That was wonderful. Thank you, Dr. Dimitri. Perfect way to start this meeting. I've been amazed at the number, but also the complexity of the questions we're being asked. We have got some very, very scientific people in this audience who are absolutely right on the button about thinking about this outbreak. So that is brilliant. We're getting to people that want to know what's happening. Well, I reckon you've got, I should have said when I introduced everybody, I'm a useless host. I'm so much more used to being the speaker, not the host. I need to learn. We thought we'd give everybody kind of 20 minutes, and Dr. Dimitri's been a little bit short, so I think we've got 6 minutes more questions for Dr. Dimitri before we go along to Dr. Christine. So here's a question from Walter Lowe. Any cat with FCoV23 infection who is symptomatic? If so, what is the percentage? So do you have an idea yet of what % of cats that are infected get sick or not?
Yeah, we do have an indication, yes, not yet documented it so well in order to have a certain picture, a certain way of thinking. We have an indication of 30 to 40%, like the infectious rate. That is coming from people that were treating colony cats, and that was the impression that they got at the first few months that we were not knowing what we do at those times. It was the first 2 or 3 months of the outbreak, I suppose. That's what we have so far.
Thank you. Based on the research where Lagavrio, it's another one, it's interesting for me to say, based on where you got Lagavrio and the EIDD medicines, based on which research, good question, were Lagavrio and EIDD approved? How did we get the approvals?
So the approval, it wasn't based, it was based on the data that we have collected from the Australian colleagues that they have researched a bit more about Molnupiravir. Actually, that was the most important research that we have, based on which they have approved Lagevrio, and that's what we used as proof of using Molnupiravir and AIDD. AIDD is not yet approved in Cyprus, but Molnupiravir is.
Yeah, EIDD 1931 is approved in the UK and Australia and New Zealand. As far as I know, it doesn't have an approval elsewhere yet. And of course, it's the breakdown product of Molnupiravir, the same way as GS441524 is the breakdown product of Remdesivir, just in case anybody was there getting a bit, what drugs are they talking about? Are there policies right now to start looking for new mutants in surrounding countries in Europe in order to track the spread of the virus out of Cyprus? Are there policies right now to prevent the spread from Cyprus?
To prevent, yes. There is only one documentation so far of finding via PCR this FCoV23 in another country, and that's unfortunately the UK. And that was reported so far. There are policies, there are suggestions that we can't enforce policies because this disease is not a notifiable disease. So there are no legal measures to do anything about that. So there are only suggestions. Those suggestions are for the antigenic testing of FCoV before any traveling to another country, quarantine for a few weeks, and then retest. That's what the suggests so far. This is what the scientific team has suggested, and maybe Dr. Liraki will say a few things about that in a later stage.
Okay, here's one. Excellent talk. Can you please elaborate a bit further on the molecular makeup of the virus in Cyprus? I think we're going to cover that with Christine's. But
Yes,
then
it will be
how are
covered
the
from Dr. Christine.
Yeah, exactly. I figure we'll let her do that. That's very much her baby. She's worked the magic there. And then how can clinicians? Where's it gone? It's moved. Hang on seconds. It's run away. There it is. Oh, it moves. That's quite interesting. Okay. Got to be quick at holding the thing, otherwise it runs away. It's disappeared now. I think some colleagues are answering these as well, which is brilliant. It was, how do you decide when the PCR is offered? So which cats are tested to give you your statistics?
Which cats are tested? Yes. So the first clinical appearance, the most common clinical appearance of FIP, as you all know, is the wet form. So mainly in the first few months of this outbreak, the clinical appearance was the wet form. So that builds body fluid in the abdomen that was sent for PCR. This is the easiest way. Some colleagues, when they have a neurological cases, they sent CSF fluid for PCR testing as well. If I understood correctly, that was the question, right? That was the question. So this is why we tested with PCR. It was offered through the laboratory that I mentioned before.
And then I think we will stop there with your questions, Dr. Dmitry, but we've got plenty of time for more questions. We'll just let you off the hook for a few minutes while we have the other talks. But I must correct my error. I am so sorry, Emma. So Emma from Bova has corrected me. EIDD has just become available and legal in Greece. So we've got
and I have to say that we were working on making it legal, on getting the license in Cyprus as well.
Perfect. And as good a time now as any, we have to thank Bova. The work they have done at getting us access to these drugs is phenomenal, because I know they have not been doing it, the work to So much work has gone into it. So thank you, Bova. Okay, Dr. Dimitri, thank you. If you would like to stop sharing your screen, I would like Dr. Christine to step forward.
Yes, hello.
Great. So what I've realized is I haven't actually introduced you properly. So, because you see, I'm not used to doing this very well. So, Dr. Dimitri, I'm going to have you introduce yourself properly at the beginning of the end question and answer, and I'm going to have the lovely Dr. Christine introduce herself.
Brilliant. I hope you can actually see my slides, and I'm just going to
Well, we can see you at the moment.
Okay. There we go. It wanted a confirmation from me. That's just always the case. Brilliant. There's no webinar without slight technical hiccups, of course. So I'm glad to be here today to present to you about this new FCoV23 outbreak. I'm Dr. Christine Tate-Burkhard. I'm a group leader or an associate professor at the University of Edinburgh's Roslin Institute, and I'm primarily focused on how viruses interact with their hosts, and have been very focused on coronavirus and related viruses for the past 16 years or something. So far ahead of SARS-CoV-2. And whilst nowadays we don't have to introduce coronaviruses anymore, actually we still have to introduce the fact that coronaviruses are actually very, very important in both livestock, wild animals and companion animals. And as we go into feline coronaviruses in general, I cannot omit this very slightly confusing slide, brilliant, of how feline and canine coronaviruses, as well as one of the porcine coronaviruses, are very intimately interlinked. Now, coronaviruses are very, very big. They have 30,000 letters in their genetic letter code. Now, contrast to other viruses, they actually don't make that many mistakes in copying their genomic makeup, but because they have such a big genome, even a small number of mistakes that they make change their genome reasonably quickly. But they have another tool in their toolbox, which is exchanging genetic information with other related coronaviruses. And that's exactly what feline coronavirus and canine coronavirus have done for decades, if not centuries. And we have the serotype 1s, canine coronaviruses, we have mixes of the two, we have the serotype 2, which is again a mixture of canine and feline, and then somewhere out of the serotype 2s, we got to the PIC coronaviruses as well. So this is very confusing, and it's an evolving picture. It's evolution in action, as many, many viruses do. So let's dive into feline coronavirus and start with a big question of why are feline coronaviruses both benign and pathogenic? And that's where we get into the so-called internal mutation theory, or some people may have heard the word biotype, where feline coronaviruses are usually enteric. They cause, if anything, mild diarrhea, and in many cases, in many cats, that's all they will do. However, in some cats, they acquire mutations, and suddenly that changes the lock, the key that unlocks cell entry. It's the surface protein called spike. It is allegedly acquiring mutations, and suddenly the virus manages to infect cells of the immune system. Obviously, the immune system is very important for us to keep barriers up, but cells also very good at reacting strongly to infections. And once you start infecting the immune cells, usually havoc happens and your immune system goes a bit over the top. And that's pretty much what makes feline coronaviruses, when they go to this mutation stage, go into feline infectious peritonitis and become very dangerous. Now, you have heard the highlights from Dimitris already, that we see a beautiful epidemiological transmission picture. You see how this virus managed to spread through different regions of Cyprus in almost like a wave. We see a very high median age, not what we normally see, that FIP cats are basically kittens. And we also see basically twice as many neurological cases. What is even more worrying when we look at the immunohistochemical staining is that we see an awful lot of infected cells in the colon. This is the colon of an FCoV23 infected cat. As a comparator, this is the colon of an FIPV1 infected cat. And you see that there's barely any virus infected cells here, whereas there's lots of virus infected cells in an FCoV23 cat, which indicates that there is likely, and we've seen that in RTQ PCRs, a lot of viral genomic information, at least, in the feces. So this is alarming. It indicates that there could be an orophycal shedding route, and we needed to find more information. And one of the ways to get more information about viruses is to look at their genetic code makeup. But the problem about feline coronaviruses is that there's generally not that much sequence information of the entire virus out there, and they have very little information about whether they are feline enterocoronavirus, feline infectious peritonitis virus. So it's very difficult to work with those data that are already there, and they are very diverse. So we opted for a pathway of sequencing that has been used very, very widely in the SARS-CoV-2 outbreak. So we can take different samples from cats, and these are the samples that are also used for PCR diagnostics. They're either fecal samples, effusion fluid, blood samples, or cerebrospinal fluid. It's good to have whole blood samples here because then you have also immune cells in there, but both a buffer coat and sometimes serum can work reasonably well. We then extract virus from these samples, or more specifically, the genetic material of viruses. But because this is RNA, we go through a translation step, basically to make it more stable, to turn it into DNA. We then amplify it because we usually don't have a lot of virus genetic material in the samples, to then read out the genetic letter code in a small sequencer, a minion, not a minion, sadly. And then we bash it with a hammer on our computers, luckily not only figuratively and not literally, and get a genetic code out of that. To do this, we split the genome up into small pieces, so about 35 pieces, just to get more detailed information. And that's where we got the first alarm bell. When we looked at our sequence pieces, most things aligned to feline coronavirus, but the spike gene immediately flagged up as canine coronavirus, strain CB05. What is CB05? CB05 is also known as a pantropic canine coronavirus. It is a hypervirulent strain of this canine coronavirus 2A serotype, and it was first isolated from dead pups in Italy in 2005. There were further outbreaks reported in Italy, Greece and Belgium in the early kind of 2000 until 2010, and it kept being picked up in dogs in different countries, including France, Hungary, Italy, Greece, the Netherlands and Belgium. And it's all in dogs that died of systemic disease. And the experimental infection shows that it spreads to a lot of internal organs, this virus. It goes to the lungs, the small and large intestine, the lymph nodes, the spleen, the liver, and the kidney, which is very reminiscent of a feline infectious peritonitis infection that we see in cats. And it's also very prominent in the intestinal content. So this very much matched the clinical picture that we saw in the cats already. And when we looked into more details and looked at basically areas before spike, areas in spike, and areas after spike, it confirmed that the Cyprus cats had the area before spike was still cat, the area after spike was still cat, but the area of spike is all in a dog spike sequence genetic. We could confirm this with what is called a bootstrap analysis, where you basically look at which area is closer to the cat sequence and which one is closer to the dog sequence. And that's ending us up with this genetic blueprint picture that most of this virus is adapted to cats, but it has obtained a new key. It has obtained a new spike glycoprotein, and actually this glycoprotein also comes in two different versions. One of the versions has a very small deletion and the other one is a full length, and I'll go into a bit more detail about that spike deletion in a minute. But what is very interesting as well, when we go back to here, when we compare the genetic code of the Cyprus cats, it's so identical that anything other than direct transmission in these FIP cases is highly unlikely. So it is very likely that this virus transmits in a very pathogenic form from cat to cat. When we looked at the actual spike sequence, we couldn't find any of the common denominators that are talking about a biotype change or that internal mutation change. But what we found is indeed that deletion. We have not yet seen any increase of the deletion in the population, but there are indications that this deletion changes the properties of spikes slightly, and it could potentially be advantageous, and I'll go into what that could mean in my summary slide. But the biggest challenge that we have been facing is that we have seen suspect cases and real cases, one confirmed so far in the UK. The first one was a very alarming one because the cat had travelled through 2 different multi-cat households before being resident in a multi-cat household. Luckily, this cat was confirmed as a normal FIPB1. The second cat in the confirmed case is actually much less risky to the whole cat population because it only went into a 2-cat household but was kept separate from the resident cat before being diagnosed with FIP and whilst being treated, obviously. But she also had no outdoor access, and the owner was incredibly cooperative, and the kitten could be treated with GS441524 and is monitored currently, but she is virus negative in all of our samples so far. So good news for kitten and good news for the UK, I would say as well. But we see that this virus is much faster spreading in its FIP form than we have seen before. We have seen fast spreading of feline enteric coronavirus, but not in this pathogenic version. It seems to be indiscriminate of age, and that indicates that previous immune status is probably not very important in this infection. And we see that it's a recombinant between FIPV1 and canine coronavirus, and the spike properties or the infection properties, the spread to the internal organs that we see, again speak to this virus being pathogenic in the form that it's in without needing to acquire more mutations. We see highly similar sequences that are indicative of direct transmission, and that is underlined by the strong antibody staining and actually the high viral genetic code load in the feces. We're about to check whether there's actually infectious virus in there, but I would be very surprised if not. We also see an increase in neurological cases, and that is very concerning. We still don't quite know how many cats are asymptomatic, and maybe we can discuss that again with Dr. Dimitris of the number of cases, because we have seen very varying numbers. And the lack of domain 0 could potentially, that deletion mutant could potentially strengthen receptor binding, but also make it be less well retained, so more virus gets basically flood out of the colon, for example, which is a potential risk factor as well. So we are continuing to look into this beast and doing virus isolation. We've obviously done a lot of due diligence on making sure that we have assessed the risk of that. We have more suspect cases and clusters in the UK and more sequencing to do. But we also have first indications that we can distinctively identify feline coronavirus 23 cats by PCR, distinct from FCoV1. And that would obviously be both time and money saving in contrast to sequencing for confirmation. So with that, I shall move to my thank you slide. My team have been absolutely fantastic in picking this up quickly, especially Dr. Amanda War, but also Sarah and Marie, who've been doing a lot of the sequencing, Harlampos and Stella and Danielle, Dimitris and Maria. And of course, couldn't do this without our kittens. These are my cats, and they're actually Artemis and Hermes. And you can tell by their names, I think this was probably fate that I was to work on a cat virus in a Greek-speaking country. So thank you very much, and very happy with questions.
Absolutely wonderful, thank you. And I love that your cats are Dimitri and Artemis. Yeah, you jinxed yourself. All yours. Okay, we've got some very good questions for you. Could there be a genetic predisposition for infectivity in the cats in Cyprus?
Sorry, can you repeat that again? I was just briefly dealing with stopping to share my screen.
Sorry. Could there be a genetic predisposition for infectivity of cats in Cyprus?
I think less so for infectivity, but more for phenotypic or symptomatic presentation. We see this very likely also in FIPV1. I think there's some new data about to come out about that. But it's also very reminiscent of what we see in other coronaviruses. Let's not bring up SARS-CoV-2, but sadly it's a good example because we have a load of data on SARS-CoV-2. And basically the story is always the same. It is the immune response genes that are important for this, and it's actually pretty clear that it's the immune system overreacting to this viral infection, and it will be similar to SARS-CoV-2, where we also see an overreaction to an infection, that genetics are likely to play a role. Not in all cases. Again, age will be a determinant, so young kittens are probably more likely to have symptomatic disease. But genetics will play into it. I would be pretty certain of that.
And certainly if we look at the paper we just published on the 307 cats, 60% were pedigree cats, of which 30% of those babies were British Shorthairs. And interestingly, they're all blues, and very occasional whites. Almost all of them are British blues. Then we see about 18%, 17-18% ragdolls, and about 7% Maine-Coon. So that really goes with what you say, that genetics is likely to play a role. But you jinxed it. You jinxed it, Christine. You mentioned the SARS-CoV-2. So the question is, how many instances of SARS-CoV-2 and FCoV23 co-infection have been observed? Has this been looked for?
We don't have any data on that yet. We have some serum samples that could be tested for that, but The incidence of SARS-CoV-2 is there in cats, but it's not huge. And we're also oddly after the initial kind of wave of SARS-CoV-2. So it's very difficult to tell whether it contributed, but we have to remember that SARS-CoV-2 is in quite a different clade of coronavirus. It's in the beta coronaviruses. This is an alpha coronavirus. And whilst we see antibody cross-protection in the alpha coronaviruses, so even with the human common cold alpha coronavirus, of which there's 2, we haven't seen cross-protection, so the antibody cross-protection between the alphas and betas very much.
Good. I hope it stays that way. I don't want these 2 viruses to play together. Faye says, I'm wondering if looking at RT-PCR directly for the canine spike RNA would be a possibility and faster than RT-PCR. Oops, Faye, you've disappeared. Somebody put in a new question, you disappeared again. I wonder if looking for RT-PCR directly for the canine spike would be a possibility for faster RT-PCR identification of FCoV23, and are there FCoVs out there that maybe have the same canine spike protein don't cause FIP?
So 2 parts to this question. So the RT-qPCR that I presented actually looks exactly at that interface, so between the canine and the feline bit. So it is very specific to this recombinant. We have seen other similar recombinants. There has been a similar recombinant. Actually, FIPV2 is technically a very large recombinant between a canine coronavirus and the feline coronavirus. We have seen smaller recombinants in Japan, but they were highly diverse. So even though they caused local outbreaks, they never spread beyond that. What we see here is a very large spread, which is probably helped by the cat density in Cyprus, which may be good news for the UK. Our cat density is a bit lower. But generally, this virus seems to have done an awful lot of things right. And again, very, very reminiscent of what has happened in SARS-CoV-2, but also what's happened in MERS coronavirus. So it has basically all the best pieces to replicate in a cat. And then it acquired a new key spike protein to get into a different subset of cells, very likely. So it has probably done everything right to be this dangerous. And that is exactly what the next steps are to investigate.
Beautiful description, thank you. I like that, it's gained a new key. And the second part of the question is, are there FCoVs out there with the same canine spike protein that don't cause FIP?
Not that we have seen. The reality is they would probably only be picked up if they cause FIP.
Agreed. That's immunity. Picking ones that, there's a lot of questions, so I want to pick ones that are absolutely perfect for you. Yeah, can FCoV23 be detected using standard qPCR from feces, in the feces? So could we detect it using routine PCR?
Basically, yeah. I mean, I've shown you the normal RT-PCR pictures. We're optimizing the conditions for that, and obviously then we'll make them publicly available as well. It looks like it's absolutely possible to optimize it, and I've seen that there's a question about RT-LAMP in there. That's exactly what we're planning next, because the accessibility of PCR is not as widespread as the possibility to have a simple heat block for an RT-LAMP assay. So that's in the making and very important.
If I can find the LAMP one, but there's a quick one. Are the primer sequences going to be available?
Absolutely, yeah. As soon as possible. We're also working on a kind of triple version that would be distinctive for whether it's FCoV2, FCoV1, or FCoV23 in one go.
Can you say that again for me, just so that everybody's got that, because that's really important?
Yes, so we're also, yeah, it's just a triple discriminatory RTQ PCR. So it has one color for FCoV23, one color for FCoV1, and one color for FCoV2. And the FCoV2 is a tricky one because technically you can pick up FCoV23 with an FCoV2 primer at the moment, so we need to make them distinct.
Sophia asks, I also heard the researchers in Davies are trying to develop a vaccine for coronavirus. Given the nature of coronaviruses in general, do you think that this would help in eliminating the possibility of further cats developing FIP?
Oh, I think vaccination is a very important piece to this puzzle, because what we've tried in the past is to have neutralizing vaccines. And since SARS-CoV-2, we know we don't necessarily need to be neutralizing, but we also can just work on preventing severe disease. And we know with FIPV1, previous immune status is very important. That's why kittens get so severely infected. So as we have published this, we have just figured out just how much feline coronavirus vaccine work is going on out there, and I'm very hopeful that there will be a vaccine that is working soon.
I agree completely, and ultimately we really want one that's an oral one for all the strays, but it has been wonderful to see how many different people are coming forward saying, we're working on it, we're working on it, so perfect. Well, I'm supposed to stop at this point, but while you mentioned RT-LAMP, so I think I'm going to say, can the RT-LAMP tests be used to improve FCoV testing access, better affordability, rapidity, etc.? And then we'll switch to Maria's talk after you, if that's okay. But that one definitely needs answering.
Oh, yeah, absolutely. RT-LAMP is brilliant because it's both off license and very affordable to do on site. And I've actually worked on a project where we've done this in the Philippines for pig diagnostics, and it's highly successful, and it creates local qualifying jobs as well in most cases. So it is a very good way to actually roll out diagnostics more widely and also into very low equipment setting. So you only need a heat block, and in most cases you can do a visual colorimetric yes no answer. You could also quantify it, and you can make it a simple phone app to do that, but it is very straightforward.
It sounds amazing. It really is thing of the future, which is great. That's what we need, simple diagnostics that are very accurate. Thank you, Christine. I know you'll be waiting and coming back for more questions. Thank you.
Thank you.
Thank you. And then our last speaker on the panel is Dr. Maria Liraki. I'm going to let her introduce herself as well, but I'm going to finish. She's just about to become one of our PhD students as well. So I love the way we're gathering these people in and saying, come on, we want to help you here. So Maria, the floor is yours.
Hello, everyone. My name is Maria Liraki, and I'm a medicine specialist. I'm based in Greece, where I see referral cases, and I have been doing for quite some time advice on referral cases in Cyprus as well. Thank you very much, EveryCat, for hosting and helping our cause and supporting research in cats worldwide. You're really doing a great job, and you're so important to us. I was approached just about a year ago by Dr. Epaminondas and Dr. Haralapos Atipa, who are key leaders in this work, to be faced with sort of a massive spike in cases of FIP in Cyprus. And I have been trying to help with all the clinical aspects that we have faced. I have chosen this picture on the left for my front slide, just to show a very characteristic lifestyle of cats in Cyprus, where they're in-betweeners. They live mostly outdoors or in cat shelters in really dense population colonies, and they're fed by sort of the so many cat caregivers in Cyprus. It is a really key feature of this outbreak, the cat density that we're facing. Apologies, a small hiccup. I'm changing the slide. My computer is slightly struggling to change the slide.
It's a beautiful picture. We're happy to like that's
It's a bit slow to load. Apologies for the delay.
Dr. Lyraki, do you have the controls down at the bottom of the
Yes.
slide itself?
Like my computer is slightly struggling. Give me 1 second. Let me just stop share for a second to give it a moment and try again. It looks like when I'm sharing, it's crashing a little bit. Apologies for the delay.
Take a little pressure off you to give you a chance to get it sorted. Shall I give Christine some questions? Because there are plenty, and I figured, is that all right? And that just gives you time.
Fine with me. I was also just going to pull up Maria's presentation. Worst case, I can share it for her and
I
she
was
can do
actually
the usual
thinking-
joyous. Next slide, please.
If hers doesn't work, then we can do that. If yours doesn't work, then I think we've all got them all, so that's good. First question for you, is RT-LAMP already available?
Unfortunately, not. A lot on the to-do list, but it's definitely within the next month or so that we're hoping to get at least preliminary working conditions. And the reality is with diagnostics, you want to make sure that they're not picking up something wrong. So my guess is 2 months to 3 months is probably the best timeline I can give on that.
You know who's working on it?
I am, well, I've put it into the
It was in
Centre for Excellence, but we're already starting because it's so important. An extra pair of hands would speed up things enormously.
Perfect. Thank you, Christine, for stepping in. Dr. Maria, are you happy to play? You are. Perfect.
I'm going to be working now. Sorry, my computer is slightly struggling today. So here's the summary. I hope you can all see the second slide. Good.
Christine, you're quicker at computers than me. You're probably already doing it, aren't you? Could you load up Maria's talk for us? Thank you.
Can you see it?
Yes, let's do it that way. Thank you, everyone, for your patience. Perfect. So we can go to the next slide. Thank you so much, Christine, for that. So just a very brief summary. We're going to go through the clinical characteristics that we have seen in this outbreak and the many, many diagnostic and therapeutic challenges we have faced, and the treatment options and the outcomes of them, and some ongoing research that is coming soon. We can go to the next slide. Regarding the clinical characteristics, FCoV23 outbreak FIP shares quite a few similarities to the traditional FIP, but quite a lot of distinctive features as well. So one thing that came out really, really obviously was that cats of all ages are affected, while in contrast to what we're used with traditional FIP, where usually kittens and quite elderly cats are mostly affected. The other interesting feature of an outbreak is that indoor-only cats were also affected, which raised initially the concern, is that a virus that has been there for quite much longer and it came out now, or is it that people are working as fomites and they're bringing the virus with their equipment and their clothes? But certainly that's something really interesting. The common clinical signs are quite similar to what we see with the traditional FIP, such as ascites, jaundice, pyrexia, weight loss, ataxia, similar to what we've been seeing. And as we see with the cat on the right, a very typical FIP cat with a distended abdomen from ascites, muscle wastage, poor coat quality, a thin and poorly cut. We can change the slide. And then regarding the clinical characteristics, the most common form, similarly to the traditional FIP, is effusive. We can see a jaundice FIP, FCoV23 FIP cat on the right. But what actually comes up really interesting is that a much, much higher prevalence of neurological form of FIP with FCoV23, up to 40% on the preliminary data we are actually looking at the moment, which is a lot more frequent than what we were used to. And that is really unlucky because most of us know that neurological FIP is much more difficult to diagnose and much more difficult to treat with much higher doses of antivirals. And the relapses were also mostly neurological, and then sometimes in the dry form, which is also a quite interesting feature of the FCoV23. We can move to the next slide. And then when it comes to the diagnosis, for the cases that we had secure diagnosis, which means that we managed to send immunohistology for the FCoV antigen, or we managed to do a quantification PCR, we have demonstrated a quite heavy viral load, which means that the virus is really, really virulent and goes in really, really high loads in tissue. And that is in contrast to what we see with the traditional FIP. And most of us know with traditional FIP that even sort of a very, very light load is a diagnosis for us because we don't get very heavy loads in most cases, but that is a really virulent case with Cyprus. And then, unfortunately, though, many cases were neurological, I said up to 40% with some of our data. And in these cases, secure diagnosis was either very difficult or really, really expensive, and the owners were declining the options. And then, unfortunately, as soon as the sort of outbreak became really viral and really, really famous, people started treatment trials without going to the vets, without securing diagnosis, and that made it really difficult to follow up who's relapsing and who's actually not being treated for the right disease. And the other feature, because FIP, most of us know that has some distinct features in blood work, that they're not sort of a certain diagnosis, but increase our suspicion for the disease. And two of them, the total globulins, often are normal in the FCoV23 outbreak. That was really interesting. And the other useful acute phase protein, the alpha-1 glycoprotein, unfortunately is not commercially in Cyprus and is not commercially available in the labs in Germany that we were sending from Cyprus. So we did not have that tool. We can move to the next slide. And this is one of the reasons we had to reach for alternative acute phase proteins. And the first one that came up, because we have quite a lot of literature that it is a useful positive acute phase protein in cats, is the serum amyloid A. And it does increase rapidly and dramatically at diagnosis and at relapses. And it normalizes extremely rapidly after starting treatment. This is what we've seen with FCoV23. And these two features would mean that this is a very useful marker for identifying relapses and for ensuring that our treatment is the right one when we don't have a secure diagnosis. But unfortunately, it is really not specific. And all the acute phase proteins are not specific. They can go up with all kinds of inflammations, but serum amyloid A more than the others. So gingivostomatitis, toxoplasmosis, cholangitis, and most inflammatory conditions in cats, we do see similar increases. So it is really important not to base our diagnosis and sort of treatment just on serum amyloid A, but it has been a useful tool. We can move to the next slide.
I have moved, but I'm not sure whether you can see the slide yet.
Yes, perfect. Now I can see. Thank you. And then next to the serum amyloid A, we actually found a lot of use on serum electrophoresis. What we found and was really interesting was that serum electrophoresis was abnormal in all the FCoV FIP cases so far, even in light of normal total globulins. And all the globulins can be affected, but the gamma alone or gamma and alpha globulin increase is more common. Now, serum electrophoresis has been a quite useful tool because of these two unique features to guide duration of therapy and doses. What has been really interesting is that it takes much longer, certainly a lot longer than serum amyloid A to normalize, 3 to 8 weeks. And what we need to figure out with our work and therapeutic drug monitoring is that because of the disease process, and would that mean that we truly are doing the best to treat these cats for very long, or is it that we're underdosing those cats, and that's why they are quite delayed to normalize the serum electrophoresis? We can move to the next slide. And for those who may not be familiar with serum electrophoresis interpretation, here is an example of a cat, an FIP cat with serum electrophoresis. And what serum electrophoresis is doing is it separates all the proteins, and we can do that in serum or body fluids, such as ascites, into multiple fractions, so we can actually see where the increase is happening. And it can do that with numbers, but it can do that with the electrophoretogram, which is basically a chart with all the proteins plotted and spread across, so we can visualize which proteins peak. And the alpha 1 and alpha 2 proteins is where acute phase reactants such as haptoglobin would fit, and the beta 1 and beta 2 globulins would be where fibrinogen, transferrin, the complement would sit, and then the gamma is where all the immunoglobulins, so IgG, IgA, and IgM would be, and is a distinct feature of FIP as we know it, to absolutely stimulate the immunoglobulins as part of the immune response. So we see an increase at diagnosis and a marked improve during treatment, although the chart looks much, much better actually in individual numbers. Gamma globulins were still increased, and we worked further on normalizing them before stopping treatment. We can move to the next slide. And then when it comes to therapy, we did face a lot of therapeutic challenges, as the poor FIP cat on the right shows, being stray and outdoor cat. And it has been really difficult to treat this high number of cases, and especially compliance for 84 days for a stray or shelter cat has been tremendously difficult. The population of strays is very large. GS 44, 15, 24 and remdesivir are absolutely a first line, are the drugs we know we have the most literature about, and we wanted to use as first line, but they were very expensive for most people. Initially, we did not have them for the start of the outbreak in legal form. They were just illegally used, and now we have them through BOVA, and we're very lucky for that. They're very expensive for most people, and of course, it was really difficult to document correct treatment and relapses early on with the cats being outdoor or living in dense populations. Can move to the next slide. And this is where Molnupiravir, the need for a new antiviral treatment, came up. And we chose Molnupiravir, first of all, because it was available in Cyprus initially for COVID-19 in humans, but that has come off the market for that purpose now and is only being used for the FIP in Cyprus, the FCoV23. We knew that the active metabolite, EIDD1931 of Molnupiravir, is effective against feline coronaviruses because it increases the mutation rate of the virus, and it was significantly less expensive than the GS 441524. So a potential interesting drug to use. We can move to the next slide. And we had some data. We had a lot of anecdotal data, I have to say. A lot of FIP specialists around the world, and especially Australia, came up to give us number. They've been using Molnupiravir for quite some time. But if we look to what we have in the published literature, it has been effective in vitro cell cultures. It has been effective we have 2 papers published on the right. The top one, 18 cats with feline infectious peritonitis. 14 out of the 18 responded and did not relapse within a few months of follow-up. And the 4 that did not respond died within the very first few days. So we didn't even know if that was a failure to respond to treatment, or these were the very, very sick cats that would die anyway. And then the second paper on the bottom right was an owner-based survey about 30 cats. 28 of them responded to Molnupiravir, and that was sort of using the unlicensed and illegal form of Molnupiravir. And there's also been sort of a trial and online available data from 286 cats. This data is coming from the unlicensed and illegal sellers, so it is by no means peer-reviewed or published in scientific research, but it does indicate that Molnupiravir has been used for quite long in cats with FIP, so it wasn't really a drug completely out of the blue that we used. We can move to the next slide. Having that data and struggling really with the unlicensed and illegal products in Cyprus, the veterinary and the authorities in Cyprus did an amazing work to authorize a special license and a special really low price just for the FCoV23 outbreak in Cyprus for Molnupiravir. And our role as clinicians was to really document the safety and efficacy with clinical questionnaires. And also we managed to secure a collaboration with the micro laboratory in Oxford and Steve Cook, who's been doing an amazing work on measuring EIDD 1931 in the serum of those cats, just to make sure that they absorb it works, and is not toxic, and it is safe for them. We can move to the next slide. And what we have seen so far with Molnupiravir, more than 450 cats have been treated in Cyprus, and we know roughly numbers like that because the stock of all these Lagevrio products has been in the veterinary authorities, and they collect it from there, so we can kind of document who is getting the drug, and it has been shown really good efficacy. We have absolutely treated the vast majority of cats with this drug. We have seen some relapse and some poor responders. They're very few, I have to say, and most of them going back, they were either underdosed or the compliance has not been great because they have been coming from cat shelters, or they have been using partially the illegal sort of drugs. The doses that we have been using extrapolated from the very little published data, but also from what our colleagues in Australia have been using. The 10 milligrams per kilogram twice a day for the wet form, 12 milligrams per kilogram twice daily for the dry, and then up to 15 milligrams per kilogram twice daily for the neurological and ocular FIP. The blood-brain barrier absolutely needs increased doses. We can move to the next slide. But we do have some concerns, and it's absolutely important that we raise those. We really don't know the best dose scheme, and we really don't have pharmacokinetics. In vitro, and that's been really interesting, there is a lot more toxicity with Molnupiravir at sort of lower doses in comparison to GS and Remdesivir. So the efficacy toxicity window may be small. Sort of what is really interesting is that in vivo, we really don't see that much toxicity, but what we have sort of been seeing with our cases is some cytopenias, and that has been quite predictable at high doses, so a dose-dependent cytopenia. We have seen pancytopenias, anemia, leukopenias, mainly with doses above the 20 milligrams per kilogram twice daily. That seems to be a frame, but we can see it with lower doses as well. There may be a potential risk of nephrotoxicity. It is excreted in urine, and that's something we have suspected in one case. We have also seen the occasional ALT increase, but we haven't associated that so far with specific hepatotoxicity, and it hasn't required the dose decrease so far. It promotes viral mutation by the way it works, so that could also be a concern about potentially being mutagenic or changing to a more virulent strain of the virus. That's absolutely not something we've seen yet, but it's something we need to be aware. So from our personal experience, I feel like Molnupiravir needs a closer monitoring to make sure that we're not having toxicity, but it's been quite safe so far. We can move to the next slide. And then regarding what is really coming soon with our research, we're really working on understanding further the FCoV23 pathogenicity and transmission. And we really don't know yet why FCoV23 is more commonly neurological compared to the traditional one, and why exactly causes different features in clinical signs. And we absolutely need a commercial FCoV23 PCR test, as Christine very rightly said, to be able to diagnose the disease across the globe. We are investigating the FCoV23 presence in neighboring countries. We have already sent samples from Greece, Lebanon, Middle East, Turkey, to see if FCoV23 has been already there or not, and we are certainly investigating that. We absolutely need more data on Molnupiravir, and also on the new treatment option that we haven't used yet, but we have some data from Australia, and it's actually coming up, and it is already authorized in Greece, is the EIDD1931, so the actual active metabolite. We want to thank Bova for doing the best to give us treatment options for our cases. And we absolutely need to compare further the traditional FIP with the FCoV23 FIP to understand that more. We can move to the next slide. And just to finish with a few thank you, I want to say a big thank you to, of course, EveryCat for having us today, and then, of course, the key leaders in this work from the University of Edinburgh, and Christine, Danielle, of course, Haralan Bosatipa, and Dimitris Epaminondas. And we have collaborated with people across the globe, and we have now the FCoV23 Centre of Excellence team. So there is a lot of research and ongoing work to come out of this. We're very excited about that. And just for a last slide, Christine, the most important thank you. There's one last slide.
That slide is beautiful. If you can bring it back.
Yeah, and of course, the most important thank you to all the vets and cat caregivers in Cyprus. They had to deal with an enormous amount of cases. They have saved the life of so many cats. We're really grateful to them.
Yeah, apologies, my computer went a bit excited there.
It is such a shame that my computer crashed, because I had a slightly newer version of that PowerPoint, but I think it did work regardless.
I think it worked perfectly. And I think your last slide was so important, because the vast majority of us on this call could not imagine what it must be to be living in a small island, and you're seeing sick cats everywhere that are dying, and you don't know what it is. The thought of it absolutely terrifies me. So the work that Dimitri and Maria and Haralampos have done in bringing this to us, in everything that you've done on site, it is truly amazing. So thank you. And I don't think I've ever seen so many claps and thank yous and love hearts as there were coming up from your talk. There's some more now, but it was just so deep, so steep, and that is recognition of what is truly amazing. Yes, that is wonderful. That's what I want to see. Yeah, we really do. We do love what you're doing. There's quite a lot of questions for you.
That's great.
Since Molnupiravir is mutagenic, are there any concerns giving it to kittens?
Yeah. Absolutely. It has been one of our concerns from the very start. We have really not seen yet big problems, but the truth is that we really need to be able to monitor that, relapses, and be able to sequence. So we're really, really keen, and we really follow any suspicious relapses to see if we can sequence and investigate what's changed in the virus after with Dr. obviously Christine. But so far we haven't seen any issues, but it's something we're actively monitoring, because mutagenic activity could be a concern. We haven't seen that yet.
My internet, for some reason, I think all the people in my flats above me must have come home and turned on the television. There's one from Frederica Scaletti. She says she's been assisting virtually in some cases in Cyprus, and several have failed to respond to GS44, but did respond to Molnupiravir or EIDD 1931. Is the mutation more difficult to treat?
That is absolutely a very great point. And what we really need to know in those cases is whether they were treated with the licensed or the unlicensed product. The problem with the unlicensed is it is really difficult to convince them to use the doses we say to them, because the people that are selling to them the drug are advising on much lower doses, and God knows how much of the product is in those formulations. So with the actual licensed GS44 of Bova, we have treated very few cats because it's super expensive in Cyprus. And with those that we've treated and we know of, they have responded quite well. But absolutely, I completely agree. And that owner-based survey that is published in Molnupiravir, quite a few of those cases failed GS and responded to Molnupiravir, but I'm wondering whether they were underdosed.
That would certainly fit with the therapeutic drug monitoring that we've been doing and now you've been doing as well. It's amazing. Some cats just really don't absorb. Certainly I can speak to GS. Some really don't absorb GS well, and others pee it out incredibly quickly, and we get very high concentrations in their urine. So you really need, and those cats are then going to need higher doses and more frequent.
But it just highlights why we need more antivirals, because a lot of people have been asking why you're pushing on new drugs. GS and remdesivir are working and they're safer. We do need more options when we have this high number of cases. We absolutely need.
And it also, if we think about how we treat HIV, it's 3 antivirals and 2 immune stimulants, and then that's perfect. So I think combinations are the way to go, because we might be able to reduce the doses and hence reduce toxicity. I'm with you completely. I think you can have a couple more questions. If the toxicity of Molnupiravir does occur, when does it typically occur during treatment? How is it usually treated? And is the treatment still resumed? Can you keep going with the treatment?
Absolutely, that's a great point. So what we have seen so far, I would probably say the first 4 weeks were a quite critical point. And then the other point was when we pushed to high doses. So we do expect some toxicity and some bone marrow suppression when we go above that 20 mg/kg mark. We've seen it at lower doses very infrequently within the first 4 weeks of treatment, but later on, the ones that we have seen were mostly pushed to high doses. And it's really variable, and we've been discussing that with our Australian colleagues that have been using Molnupiravir for many more years than we do. And it's really variable what we're doing in those situations. So when we have really mild drops in the blood counts, we potentially, depending on our dose and the clinical response, sit and wait and monitor closer. But when we have marked drops in blood counts, we have to do something about that. We start becoming nervous. So then we may drop our dose. And we had the occasional case that we changed to GS more because we were worried and we didn't want to put the cat in risk. But in the majority of cases, it was mild and we were sitting and waiting. So it really depends on the severity.
Okay. And then last one, and then we'll open up the panel. In my memory, if my memory serves me right, Molnupiravir became available more or less at the same time as Paxlovid, another drug that was treated for COVID. Are there any studies on the use of Paxlovid for the treatment of FIP?
Absolutely, that's a really, really good point. So there are no studies as far as I know, but I know that the combination of antivirals that the Paxlovid has been tried in vitro, and as no one would be surprised, it is effective. It would be interesting. There is also some in vitro toxicity associated with Paxlovid in the feline cell cultures, and that's even worse, because I said already the toxicity in vitro with Molnupiravir seemed to be worse than GS, and Paxlovid seems to be even worse. I certainly think it's not completely out of question to try that as well. But based on the toxicity issue and the availability at the moment with the human COVID-19, because currently it is the first line treatment at home for COVID-19, it would be tricky to try on, and certainly there's some toxicity issues, but it is absolutely combination antivirals is absolutely the way to go, but no data.
Agreed.
Just to add to that the problem why Paxlovid was discontinued in many countries is because it causes Paxlovid mouth. And I think in oral medication that makes cats get weird mouth taste. They will get off it in no time, and feeding them.
That is a very good point. And certainly, do you know, people have been trying it in Australia, but the problem is in so many countries, it wouldn't be legal for us to go anywhere near anyway. And then people go to the illegals and you have no idea what concentration is in them or if there's anything in there at all, whether it's all toxic things in there. So I really do ask people to stay with legal drugs if at all possible. I have to say, stick to legal unless you have no other options at all.
And that's why we went for Molnupiravir, because we just realized most of the cats were treated with illegal product, and we were just losing the opportunity to monitor that.
As you're on the screen, let's stay with you, Maria, for a second. Raymond says, could you say what the side effects are with Molnupiravir?
Yeah, absolutely. So we can see the occasional ALT increase, similarly to what we see with GS and remdesivir. And this has not yet been associated with severe hepatotoxicity to the cases that we've seen, but we're monitoring that actively to see if it's sort of meaningful or just reactive from the drug. We are monitoring for nephrotoxicity because it is excreted from the kidneys. We have suspected that in one case, but we didn't manage to be clear on whether that was the FIP or the drug, but certainly at least in one case, azotemia developed after starting treatment. And then the vast majority has been the cytopenias, the blood cell count drops that we're actively monitoring. In some cases, really, really mild and not significant clinically, but in some cases could be more severe and could change your treatment course. It has been reversible in all of the cases so far. I have been contacted very recently about some skin changes, and I wouldn't be surprised. This cat has been treated with GS and then Molnupiravir. So I wouldn't be surprised if we see skin lesions based on the literature. There's literature about faulted sort of ear tips as well, and obviously drop whiskers, which also wouldn't be a huge surprise, although it hasn't been clear because we haven't seen that very much in Cyprus yet. So it's not very clear if that's the FIP itself or the product, but it wasn't clinically significant.
And I think it's interesting that, we see 30% of cats treated with GS have got raised ALT, and yet it doesn't seem to cause them any trouble at all, and it stops as soon as the drug stops. We need to do more work on that. I love Angela's comment. How about legalizing it, guys? The illegal also brought you to this seminar. I'm not sure quite what she means by the second part of this, but I'm with you. Let's get drugs legalized. The problem is, none of these drugs have gone through long trials that you would get to have normal legalized drugs. We're talking about specials. This is a specials lab that's brought them all forward, and we're very grateful that they have, but nobody has gone through the multiple rounds of testing that you have to get normal drugs licensed. So we're really working at the same way as on the peak of an outbreak as drugs were legalized during COVID. So
we're certainly very lucky that we now have some legal options. And apart from the human Lagevrio Molnupiravir, now Bova has the EIDD 1931, and we have legal GS and remdesivir. We're very lucky, and I think Bova has helped as well a lot on that. It is really what we have obviously all realized is it is really difficult to do a clinical trial, and it always comes a lot later than the action of the outbreak. So we had to treat a lot of cats with not the amount of published data we would want it to have, but
in which case I'm going to give one more question to you, and then I'm going to let you save your voice for a minute. You will be pleased to hear, everyone that's watching, because you'll all be thinking, eek, we're all about to go offline in about 3 minutes. The team have all agreed to stay until 6 o'clock our time for you. So after that, we've got a nephew for one of our speakers who is going to run amok, and she's going to be dragged out of the room by her hair. So let's have a question. Where was it? Oh, yes. We've treated, Katrin says, treated 2 cases in Chile with Molnupiravir, and one of them, oh, hang on a sec, my cat's being stupid. Get down, you idiot. Sorry, crisis. Look on yourself. Get out. Oh. My apologies. That is a willful 2.5-year-old Maine Coon boy who still has his testicles. And he didn't like the fact that I'd been speaking to a television for too long. So he just leapt on top of the proper television, which is not very stable, and was trying to flip the whole thing over to get my attention. He got my attention. He's now got his daddy's attention, and he's been locked out of the lounge. Apologies. Right. Christine, thank you. There's quite a few for you. Did you pick up some you wanted to pick up on?
I think I managed to answer most of them that were targeted at me. I think we still had a lot of questions about drugs, so happy to stay on that. I cannot see any evident ones. The other one I think we kind of should discuss, also with Dimitris, is and it has come up earlier, is the question of how many cases are there actually? 300,000, 8,000, 10% dead, 50% dead. So what's the evidence? And I think people are curious to know, because newspaper headlines are very impactful, of course.
And very dramatic, I must say. Yeah, the numbers, not actually the actual number of cats in Cyprus, are not yet defined. So we cannot know for sure how many cats were dead. That the thing that we know for sure is the colony cats that were affected at the beginning of this outbreak had an infectious rate about 30 to 40%. So if, let's say, we had a colony of 100 cats, 30 to 40 cats were had this clinical appearance of FIP. Some of them were rushed to the vets, some were not. So the exact number of how many they were dead, it's not possible to know. But there are 2 factors that we need to consider. The first factor is that there are a lot of caregivers here in Cyprus for stray cats, which are willing to put their hand on their wallet and give money to help the cats. So that is a very encouraging thing for Cyprus cat population here, because a lot of people manage to save a lot of stray cats due to this process. And this process usually, at least at the beginning, involved illegal mitigation, as we say. We did our best in the time about 5 to 6 months to legalize and give the chance to people to use legal drugs, which are the optimum solution. As we know, as veterinarians, we have to suggest the legalized drugs because there are a lot more stable drugs and they have been used and tested. So we have done our best to do that, and we have provided solutions for the people. We cannot stop the black market, of course, we know that, having said that, we know that the black market saved a lot of stray population at the beginning. So it's a little you have to weigh things. So we don't know the exact number, but that is pretty much the answer that I can give you.
Thank you. And before I'm allowing you to take another question, I would like you to actually introduce yourself for us. A little bit late in the day, I'm being rubbish, but please say why we think you're the perfect person to be here telling us about it.
Yeah, the perfect person. I don't know if I'm the perfect person. I'm a veterinarian, of course. I am the vice president of the Pan-Cyprian Veterinary Association, and we have been involved from the very beginning to monitor and document the epidemiological data of this outbreak. and we were responsible for giving details, organizing seminars for vets and the public. We have had really good help from companies. Let's say BOVA was one of the companies to help us with the distribution of these seminars. Dr. Maria Liraki gave a free of charge webinars for vets and helped a lot with the diagnostic approach and the treatment, because we had to have a uniform approach of that in that matters. That's why I'm involved. I am because of the association, but my very was my willing power was to help the cats, because there was an alarming situation and we have to have some solutions. So I have helped to provide these solutions.
There was a question, where is it? They were asking how you initially got Molnupiravir licensed. How did you get it? How did you get it legalized?
We didn't import Molnupiravir. Molnupiravir was available for humans for COVID-19. And there was a decision of the Ministers' Cabinet, which is the high body of decision body in our constitution, that gave this permit to use this drug for this for this disease due to the necessity and the high outbreak rate of this due to the cats dying. It was something that we have asked as an association, but we have as the support. We had the support from various animal charity organizations, animal caregivers to help. They help us a lot with the political pressure to have this done. But legally, how we did it, it was through the Ministers' Cabinet. It was the president's decision, something like that.
And we are thankful for the president. Are you comfortable talking about the antibody side of feline coronaviruses? Whether or not it's valuable to look at antibodies by way of a diagnosis or monitoring. We've got a number of questions about those.
Yeah, I think that is for Maria to answer.
Then
probably.
I shall answer that to Maria. But here's one which so how do you decide on the protocol? Do you go oral versus injectable? How do you decide which is the best way to start?
Mm-hmm. That is for Maria as well. Mm-hmm. I can say a few things about at that time we needed to have something very cheap in order to treat the stray population, which was affected. A lot of the stray population was affected. So we turned to various solutions. We tried to import other drugs as well, but we didn't. That wasn't the case because we have legal problems to do that. So Maria can tell you a bit more about this information you need.
Yeah. Yes, of course.
Then I think we'd better ask Maria. Please continue.
Yes, of course. Thank you. So with regards to the oral versus injectable, we have very much been following the same ISFM guidelines that we have for GS and remdesivir as well, that as soon as cats are eating and can reliably absorb oral, we do use oral drugs because they're cheaper and also we don't have that pain associated with injection that sort of cat caregivers report that the cats have been avoiding them when they be doing the injections. They need a lot of training associated to how to do that more feline friendly. So we do prefer oral when we can, but when we have a hospitalized cat with vomiting, anorexic or dehydrated, we know that those cats will probably need a few doses injectable to absorb properly, improve, and then transition to oral. So we have pretty much decided on a case-by-case basis. We did not have anything Molnupiravir related injectable. So then that would be the remdesivir, the legal when we could, and then transition to the oral Molnupiravir at the sort of first available opportunity. So very much following that. Luckily, sort of the majority were going straight to oral, but we certainly have those cases that had needed a few injections before. And then with antibodies, we are sort of with our research, we're super interested in the antibodies. Initially with the outbreak, our main focus was to discuss with the vets that antibodies don't tell us anything about diagnosis and do not have sort of antibodies as part of the diagnosis protocol. And we were really focused on that, but sort of looking back at research is interested to see sort of high antibodies in the majority of cases, but some cats being negative at the time of diagnosis, where sort of the macrophages are having all the virus in, and the immune response of the cat is very low. So we've seen variable results. And what else is really interested, and we're looking into that, but we need some funding before we do some proper research on that, is how long they maintain antibodies for after being treated. That's really, really interesting to see the very few cases that we have followed in that aspect, because clinically we try to educate vets not to base decision on antibodies, but interestingly, they do maintain high antibodies for very long, the few ones that we have seen.
I'm sorry, I missed part of that. Busy answering questions, but I will ask you offline. There's another one. Is the cell-mediated response versus humoral response? Do we know? Do you want to give a whole tutorial on FIP, or shall we stick? Because that's. Do you want to say quickly the difference between cell-mediated and humoral responses with FIP? But let's keep it short because that is a whole lecture.
Yeah, absolutely. And that's the whole difference between sort of being asymptomatic or having the wet form or having the dry form, or the neurological, the type of immune response from each cat. So some cats do manage to mediate a cell response, and they do seem to do better with regards to disease, while some other cats will actually bring up their humoral, so their antibodies response. And those cats seem to are the ones getting more sick and getting the wet form of the FIP. So it really depends on how they respond. What is interesting is how many more are getting sick with this virus. So I don't know if, Christine, you have more to say on that from the sequencing of the virus.
I have to apologise. I've also been answering questions, so can you repeat the question, Danielle? I've listened with half an ear.
Maria, you go ahead. You repeat it. You're going to be quicker than me.
We were discussing about the cell-mediated immune response versus the humoral immune response. And we were saying that a lot of that is the genetics and the individual cat's response. And we know that some of those cats are inbred, being a stray population, and that would affect how they would respond. And the humoral responses are more likely to get the wet form and being quite sick. And I didn't know if you had any comments from the genetic side.
I think actually virology will tell us much more about this in terms of how, I mean, the indication from more neural cases are worrying, and they completely agree with the canine spike possibly infecting a much broader range of cells, and that would mean as soon as you can break down the blood-brain barrier. Or we've also discussed the potential option that has been seen in mice recently, that there's actually a direct link into the brain through the lymphoid system. Nobody has investigated that in carnivores, let alone cats and dogs. So there are potentially many more routes of infection to the neural cells that we don't yet quite understand. And we know this virus goes into the lymphoid system and could change things. The interesting bit is that the actual antibody response doesn't seem to be particularly linked to whether we have an immune overreaction or not. That's the only thing I can say, that antibody levels are probably not reflective of whether you're sick or not, in which way. And that's what all the coronavirus research tells us, and that's also what SARS-CoV-2 research actually tells us. But they hopefully do then indeed do what the vaccines do, that future severe cases are hopefully prevented. We don't know, for coronaviruses, the problem is that immunity doesn't last very long, and our best friend, oddly enough, is repeat infection or repeat vaccination. And that will be something to figure out. And that's also, I know there's another question about the kind of case loads kind of slowly going down. Dimitris can probably answer that as well, whether we have a winter spike, which we were potentially planning for. But my guess is that the virus might continue to go in waves as we see this with SARS-CoV-2, because herd immunity, which has become a swear word, but it's actually immunological reality that we reach a peak where we have enough immunity in the population that the virus kind of doesn't really spread much anymore. But that is temporary, unless you have a virus where you have completely neutralizing immunity. But sadly for coronaviruses, we don't really achieve that. So it will continue in peaks and troughs, and that's where the risk for the international cat population remains, that this is possibly not necessarily going to disappear.
I totally agree with you, Christine, on that. And I must say that adding on what you have said so far, okay, the winter time, which is starting here in Cyprus on January, actually, that's a late start on the winter, has much more dangers for the cats that are staying outside, the stray cats, because of the co-infections of other viruses and other conditions that may have a potential risk on their immune system. So that is what we were waiting to see. That is the first thing. But I totally agree with you that it will come in waves, because maybe the new kittens that are born, that are going to be on finding this virus on finding this virus, they could be infected and have the symptoms, the FIP symptoms. But we do not know how are they going to be, how is it going to be, the infection is going to be stated in these kittens, the new kittens that are born in the next few months. So that's we must continue to monitor this epidemiological data of this outbreak and the PCR positive cases, and see how this epidemiological curve is going to come, let's say.
And continuing on with that, Dr. Dimitri, the question is, do you know if there are some plans to control the stray cat population in Cyprus or anywhere else after this outbreak? So other ways of controlling the stray cats, trap neuter returns, anything similar?
Mm-hmm. There are a few ways, yes. There are a few ways. They need to be a bit more, let's say, updated, let's say. There is a trap and neuter program, a sterilization program from the government, offered from the government for the stray cats, for the stray cat population. And there are several funding organizations that do, and animal care organizations that do this control of the population. Our association is planning for a few years now to do something like that, but we need to get a little bit ahead of ourselves at some point and do that. And we will do that, but this will come in the future. The first thing that we have to do, though, is to document the complete population of cats in order to know where those cats are located, and what is the number what is the starting number of the stray cats that needs to be neutered in order to do a proper sterilization program. Having said that, we have launched an initiative here in Cyprus. Our association did that to monitor to count the cats, and we have named it actually the Count the Cats project. So it is an initiative to do that, to document the population of the stray cats in Cyprus, and that will be the starting point of our campaign.
I think that's essential. Okay, if I may, can I ask Christine a question? It's about the amounts of proteins are you finding? I'm not sure how much CSF you've had to look in yet, but the question is, what are the amounts of concentration in CSF and comparing neurological form, suspected form? I think this might be a bit of a how long is a piece of string? Have you looked at CSF to see whether or not it's useful for diagnostics?
The samples that we had from neurological cases, a lot of them were CSF, and they were equally useful and had an equal range of CTs as the. In fact, I think most of our samples from the neuronal cases were CSF, and most of the cases of the effusive form had effusion fluid as our diagnostic sample. And they had equal spreads of CT values.
Sorry, could you tell the audience what a CT value is?
Sorry, it's technically an arbitrary value that tells you how quickly the signal from a PCR comes up. It's a cycle threshold, is what it translates to, but basically it's when the positive signal from your PCR is above background, and the lower that signal is, the more virus you have present. The higher that signal is, the less virus is present, and we have seen that whole spectrum in the samples. Equally, I mean, for diagnosis, ideally samples get preserved at least at -20 degrees. The virus is a very good protection vessel. That's a good thing to have, because RNA is normally not very stable, but if it's wrapped nicely in a viral protein shell, then that's brilliant. If you have any suspect samples, I think the reality is at the moment, get in touch as soon as you can, and we'll do our best to get them assessed as quickly as we can. And we keep working on the RT-qPCR, and also to have supports from laboratories that can turn samples around much quicker even. Danielle, you're muted.
Best way to have me. Mentioning that your lab is happy to receive these samples is perfect. I think I've got time to squeeze in one or two more questions. I've got to ask Sneha, because she's one of my protege, brilliant students. She asks one for Maria. We've seen relapses after Molnupiravir. Would you then opt to increase the dose or swap to GS or remdesivir instead in such a case? And then if opting for a dose increase, how much would you recommend? And then further, how does the Molnupiravir compare to GS/remdesivir in terms of blood-brain barrier penetration? I can repeat each bit if you want to answer a bit at a time.
Yeah, absolutely. Although we don't have sort of enough information in the literature yet to support that, the tendency, based on the therapeutic drug monitoring work we have been doing, is to first increase the dose if we have the space to do it in terms of toxicity. And that's because the doses that we have been using are sort of not backed up scientifically, so we don't know the best dose schemes yet. So potentially we have seen that we have been underdosing, especially the neurological ones. So similarly to GS, we tend to try a dose increase before we would reach for another drug, although we very well know that the future may well be combinations. But so far we've tended to do dose increases. In terms of the neurological form, and it's really not fair for GS in Cyprus, because the vast majority of GS has been the illegal source, and the data that we have with the legal product is brilliant. But certainly we haven't seen any huge difference. They both seem to be quite effective with the neurological form at high doses though. So for Molnupiravir, that would mean starting with 15 mg per kg, but often having to go closer to the 20 mg per kg. And where is sort of close monitoring?
Would that be once a day or twice a day, Maria?
Twice. So Molnupiravir's pharmacokinetics that we extrapolate from people and from the anecdotal using cats, but we do confirm those so far with our therapeutic drug monitoring is twice daily. Certainly sort of the trough samples closer to the 12 hours have very, very little EIDD in them. So twice daily and closer anything between 15 to 20 if needed for the neurological ones. Nearly all of them have responded quite well, I have to say.
Thank you. Okay, now it's 6 o'clock, and unless my speakers are just desperate to take some more questions, I think I'd better close the questions, but don't worry. They have promised they will try and look at the questions for you. They're all, you can imagine, incredibly busy working with this outbreak, but they will we'll try and divide these questions up and get them to the most important people. And I'm going to add my thanks to start with to our 3 fabulous speakers. You can see that without each of these people, this outbreak is going nowhere. So Maria, Dimitri, Christine, we owe you a great deal. And EveryCat, we owe you a massive thanks, because obviously you're already funding our work. Thank you. And you know, getting the funds and the way they've brought together this fast funding cycle. So they're calling for grants on this outside their normal cycle because they know how important it is. So big thank you from me to all of you. Thank you very much.
Well,
and I will now hand to Brian.
Well, actually, Brian's had some computer issues, so you're going to be handing it off to me.
And I'll hand to Vicky.
Okay. My name is Virginia. I am the Outreach and Education Specialist at EveryCat Health Foundation. And it's my honor to thank the 4 of you for participating in this. This is one of the largest webinars we've ever hosted. Obviously, this is an issue to cat lovers everywhere. And as Dr. Gunmore mentioned, I am going to collect the questions that were not answered, and I will make them available to the speakers that they can answer when they have time, if they've got, and I will be putting those together probably in a couple of weeks into a document and sending them out to all of the attendees. So if your question didn't get answered, just be patient and we'll see if we can get it answered another way. Thank you to everyone that attended. Thank you to Dr. Vicky Thayer, who worked in the background with me to help kind of call some of the questions because there were so many. We do appreciate that. I appreciate her as well. Dr. Laraki, Dr. Epidemiological, did I get that right? Dr. Tate Burkhardt and Dr. Gunmore, I can't thank you enough for your time here. Thank you, thank you, thank you. And don't forget, your donations, this is to the audience, your donations are what help us fund this research. And we want to thank also our wonderful partner, Zen by Cat and Peter Cohen. Without him, he works tirelessly. And if you just want to look at some cool cat videos, visit Zen by Cat. All right, thank you, everyone. Thank you for attending and thank you for your help, speakers.
Thank you very much. Bye-bye.
Thank you, everyone.
Bye, everyone. Thank you.
Bye-bye. Take care, everyone.